and let's get started. I hope you can see my screen. I will just decrease the size of all other icons that are popping up, like the control panels. So right now I do not see if anybody is getting ready to talk. So even if you raise your hand, it might be difficult. So I hope Anita, who is here today on the faculty, may help me. And if there are any questions that you can simply unmute and you can jump in. That meaning that I have not asked you to does not mean that I am rude, but I simply could not see you when I'm on full screen presentation mode. So our today's topic is inspecting and what to inspect. Now, we have the definition according to the handbook right here. Inspecting is the critical appraisal by visual, olfactory, and tactile means of the capability, adequacy, and/or current performance of a physical entity, such as laboratory, testing facility, field site, equipment, for adherence to established regulatory standards. It's a bit complicated definition, and you do not need to remember it. And sometimes you may see there are overlaps if we talk about auditing or inspecting, or our organizations and OECD actually decided to keep inspection for that activity conducted by a regulatory authority, while auditing resides with the organization, both for auditing your vendor suppliers and performing internal audits. Let's not allow definitions to confuse us, and let's check what is actually hard, what should be inspected or audited or whatever you want to call it. So GLP principles define 3 types of inspections. We already covered that, but it's good to remind ourselves: study-based, facility-based, and process-based. It's quite clear what it means. Once we are focusing on a particular GLP study, at a time on the facility itself, and finally process. We follow a certain process from start to end to see whether there are gaps, so whether it works as we expected or as we assessed, and give us perhaps some hints for improvements and for identifying potential gaps that can produce non-conformities. And inspections should focus on activities which present the greatest risk. So what to do about the risk? We have 3 steps. We need to identify the critical processes and critical data where errors matter the most. Then identify and analyze risks, considering their impact, their likelihood, and our ability to detect that the risk is about to explode. And then we should focus on high-risk areas to prevent those errors that would ruin the study, derail the whole process, and so on. And as I said a couple of times, many times it's just common sense and basic logic that are giving us the best advice. If it does not make sense, if it breaks basic logic, then there is usually something really wrong. So you do not need to memorize definitions or to memorize articles, this or that, of either OECD or FDA regulations or any other that says this and that. As long as you understand the meaning, what we should do, how we should do, why we should do something certain way, that's the guidance sufficient enough to give us the right direction. So what should be inspected? Component labs. Let me also take this thing out of my side. Like chemistry, histology, pathology, clinical pathology, surgery, microbiology, and so on and so on. Non-laboratory sites, like field sites, test plots, mesocosms, simulation structures, depending on the type of the study. storage areas for both test, control, and reference materials, specimens, samples, media, feed, bedding, chemicals, reagents, equipment, supplies, and practically everything what we need to carry on a study. Computer facilities and associated control procedures with systems, including disaster recovery, would be focusing on inspecting the facilities, either our own or that of our critical vendor. Protocols, SOPs, facility records, operating permits or related information on each lab site location. Data under active collection in each laboratory or site. Labeling. Equipment, we already talked about equipment, but this is more associated with test system maintenance, like animal rooms, test pods, area, and study conduct. Maintenance itself, calibration, validation where required, archives, including retrieval and environmental controls. Lab or field site that participates in a study, including vendors, contractors, subcontractors, and storage conditions, whatever we need for the study. So there are certain technical skills and soft skills that should go hand in hand for a GLP professional to be really well equipped to do the job. And technical skills, of course, include everything related to the standards, processes, procedures, protocol, and so on. And soft skills are related to communication, problem solving, conflict management, social relationships, because it's not only what we say, it's also how we say that matters. And the way how we say something, for example, how we communicate non-conformity, may either help the auditee or inspectee to understand and be likely make improvements and comply, or create resistance or self-defensiveness, start arguing. It doesn't happen too often, but it does. It does happen. In my experience, I was actually auditing a lab, and there were some things that, in my opinion, really needed improvement in terms of their risk assessment of their vendors and suppliers. I put it as a finding, and the auditee actually was fighting back quite adamantly, saying, no, we discussed it with our management, our processes are just good. We are not going to change anything. I tried to rephrase it and somehow help make it likely to adopt, but the answer remained the same. We are not going to change anything, our processes are great. As I said, it happened once for me, so it's not very often, but you may get this kind of attitude, especially if the vendor or service provider it has a kind of dominant position in the market. This was a GLP lab, one of the most sought for labs in Europe. My client was European, so it's not going to do the analysis they needed to do in the United States or Canada or Australia. They wanted one in Europe for filing in the European Regulatory Agency. And because this company was really on the top, they did not accept that something might not be perfect. Finally, my recommendation to my client was this area exactly relates to something that you do not need for your project. So it's up to you. I don't like the attitude, but if you are willing to accept it, and if you see that, okay, it's extremely unlikely that our project is going to be anyhow affected, go with them. And finally, they did exactly that. They said, we don't like it either, but as you said, it does not present any risk for our particular project, which is on the top of that short time. So This is a for me as an independent auditor, that's my client decision. I give my recommendations, but if you are part of an organization, then you cannot really take it the same way. You cannot say, let's say, if you get a position from the study director, or you get a position from the executive management, you really need a lot of social skills and negotiation and diplomacy to help your organization to be compliant. You cannot just let it go, think like I tried 3 times, I got 3 no's, and that's the end of the day. If something happens, then that will be part of your responsibility, although not legal responsibility, such in the GMP area, where the QA and the general management or the CEO of the organization, they are liable in front of the law. So you won't go to jail, but still there might be consequences. And what is the goal of all the GLP work? Well, first of all, of course, we want to have happy clients and stable portfolio. We want to have a good position on the market, good reputation. That's very legitimate. But there is something more. The work that we do in GLP, the results are taken for decisions for the drug development and life cycle, whether it's stability data, whether it's data before the first in-man study. And this data should be robust, reliable, and it should help making informed, well-grounded decisions. So it's not just activity to have a good business. Of course, that's desirable, but it's not the only thing. We need to see a little bit further. We need to see, okay, what is our output used for? What is the impact? And what if what we do is not done properly? What kind of consequences can it have downstream? Now let's move to the questions. Let me try to take this completely out of sight because this control panel is simply blocking my view. Give me a second and apologies for any inconvenience. It's a little bit bulky on the screen. Let's see how it goes. Yes, better. So we have now study 456, and it requires testing to be conducted in compliance with 21 CFR Part 58, meaning FDA, according to its protocol. And we have lab, let's say the shiny new lab, that performed the pathology portion of the study. The lab uses one procedure for processing and preparation of slides, and the procedure is repetitive in nature. The QA statement in the pathology report shows that a process-based inspection was performed to monitor GLP compliance during the conduct of the pathology portion of a study. With which of the following did lab fail to comply? So what was not done well? And your task is to select one. Nothing, as it is the test facility and not the test site that is responsible to have its quality assurance unit perform critical phase inspections. Nothing, as process-based inspection is valid for procedures used that are repetitive in nature. Perform critical phase inspection for work done on study 456. And finally, amend the protocol to specify that the QAU will do process-based inspections during the conduct of the pathology portion of the study. I can't see the chat now, because if I put it there, it will just sit in the middle of my screen. But hopefully Anita can tell me what kind of responses are coming. So we have 1, 2, 3, or 4. Only one is the answer that should be chosen.
So far, we have all 3, whoever answered it.
Okay.
Yeah,
so we have
still coming up. Oh, someone said 2.
Okay. So no prevalence, no pattern, no unified opinion this time?
Yeah, it looks like mostly 3, and then we have 1, 2.
Okay. Let's give it 2 more seconds for those who haven't decided yet to have a chance to put their preferred answer. And let's take a look what the answer is. Yes, perform critical phase inspection for work done on study 456. Exactly. That's what GLP says should be done. And since the internal inspection was focusing only on one part, definitely there were other areas that should be looked at as well. Let's move forward. The
Do you
QA
want to discuss number 2? Someone had number 2, so we can maybe discuss why 3 was better than 2.
Okay. Anyone who chose 3 or 2 would give the reasons for your choice. Just unmute and say that.
Rebecca, do you feel comfortable talking about why you chose 2?
Can you hear me okay?
Yes.
Oh, fantastic. I was just thinking about one of the previous slides where GLP inspections can be a process-based inspection. So I guess I was assuming that there was a previous process-based inspection for this process.
Kimala, do you want to go ahead?
I could not hear very clearly. The audio was a kind of difficult. But if I understand correctly, there was more of a misunderstanding, right?
Well, she was saying that, Rebecca, please correct me if I'm wrong. She was saying that she thought process-based inspection still is an inspection. So that's why she didn't think that 2 would be totally incorrect.
Okay.
Yes, please correct me if I'm wrong, Rebecca.
That's correct. Thank you so much.
Thank you. Anyone supporting other opinion, like 4 or 1?
Nope, just
No.
all 3 and then 1, 2.
Okay, so
hi, Kamala. Kamala, sorry to interrupt. This is
Hi,
Jyoti. How are you?
I'm fine. How are you, Jyoti?
I'm good. I'm good. So just one point from my side, that probably the pathology portion, we consider it as a critical phase. So as per by the regulations, the critical phase definitely needs to be inspected by the quality assurance. So maybe that's the reason. I agree it's a process-based inspection. I mean, it could be a part, but for when we talk about the study, it is considered as a critical phase. So it should be audited independently as a study-specific inspection.
Thank you. Good point. Let's move to the next question. If there is no one who would like to contribute to the discussion on this one. So this is about SOPs. The quality assurance SOP for inspection states that QA will select 1 or more in-process phases of each study to inspect, depending on the length of the study. A study director tells the QA manager that the SOP should be revised because in-process inspections should only be performed if requested by the study director. What would the most appropriate response be? We already had this once, but perhaps not everybody was here last time. And sometimes we may have overlap in the topics. So what is your vote? The SOP will not be changed because QA is required to inspect each study at intervals adequate to assure the integrity of the study and maintain written records of such inspections. 2, the SOP will be changed because the study director is the single point of control for studies, and if the preference is to have QA do in-process inspections only, on a request from the study director, that will become the new procedure. 3, the SOP may be changed, but QA will have to check with test facility management first to see if they agree with the request, as it would affect all studies in the facility. And finally, 4, the SOP will not be changed because QA auditors need to interact with the technical staff so that when data comes to QA for review, they understand how the data was collected. So
everybody has selected number 1. There's no, nobody wants to argue that.
And everybody is right. Yes. One nice thing is what the study director wishes, but another thing is also the QA responsibility required and mandated by GLP, and that is very clear. You may argue that in real life, we may end up somewhere 3, but hopefully then the management would understand the responsibility and will support the QA and will say, no, QA needs to fulfill their GLP responsibilities, and we are not going to change anything. I hope that would be the answer.
Yes, someone posted 5835B3, which I don't have it memorized, and I don't have my book by me. But
okay, that's great. Surprisingly, there was no regulatory reference in the answers provided in the exam.
yeah,
In the
I think we
test.
did that last time. If I remember correctly, we did that last time, but not this time.
Yeah, so thank you anyway for pointing that out. I'm not good in memorizing the paragraphs and articles and all that. The important thing is just by thinking about it, even without the exact reference, your common sense and logic would guide you, as we just saw, to the correct answer. Next one. To avoid contamination, separate areas shall be maintained. Yeah, you will say for everything, but the most important, reagents, chemicals used for bioanalytical analysis, disposal of the test and control articles, automatic pipettes of various volumes, mixing of the test and control articles with a carrier. And that should be very easy again to answer. I don't expect too much of a variety.
Yeah, I think we had this answer before also. So
Yes.
all answers are 4.
And this is correct. Yes, of course, you would not mix reagents and chemicals together and have them sitting next to each other, especially some of these, depending on the packaging and sensitivity, and some of them might need lower temperature than the other. So you would not put everything under one hot roof. Disposal, may say, does it really matter when we dispose something? The important thing is to have a safe disposal, first of all. And then, yes, if I'm sitting in the lab, I don't want to keep checking each time if this is the correct pipette I am reaching out, and I would arrange them in a way practical for me to work. But the worst thing that can happen if there is a mix-up of the test and control articles, and suddenly we do not know what is in, what are we going to give the animals. I have no idea, and I need to start over again.
I think we missed this. There you go. Okay.
And we have the regulatory reference this time. So indeed, it's the mixing of test and control articles with a carrier. If you have any experience with the GMP world, also there, the mixing of different ingredients, different of active substances, any kind of mix-up in GMP, it's the thing that is dreaded and has to be prevented. And here it's very similar, and it's actually the same in GCP world, where if you have patients who are in a double-blind study, and suddenly there is a mix-up, and patient in one arm gets the product from the for the other arm, and so on. Even in bioequivalence, if we have mix-ups, it's always a disaster, and it's visible. It's visible from the analysis because suddenly you get nonsense results. I remember again a case, the client, they came to me that it was a bioequivalence PK study. They came with the PK results, and they somehow looked like total mix-up. So what I did, I put it in an Excel sheet and I visualized it. And normally you would expect a nice, neat curve that would go the expected way. And it looks something like, you know, zigzag. And finally, the only explanation was there must have been a problem either with a labeling of the samples, and they were incorrectly labeled when they were sent to the lab, and the correct time points were attached to wrong vials, or it happened at the lab when they were feeding the EHPL's TC. So finally, further investigation showed it happened in the lab where there was a new technician, and somehow the second one who usually was expected to guide this person was not there, and he just put randomly the samples. And for bioequivalence, that's a disaster because regulations prohibit repeating the analysis. So my client unfortunately ended up with a completely ruined study. Test substance characterization is the next one. Test facility conducts a GLP test substance characterization study. The analytical reference standard is furnished by a supplier with a certificate of analysis confirming identity and purity. The study director conducts the study and completes the report with the reference standard as supplied. Considering the GLP requirements for test substance characterization, which best describes the most appropriate course of action? Again, just one. Analytical reference standards do not require characterization prior to their use in a study. So there's nothing, no issue. The lack of a GLP characterized analytical reference standard must be included as an exception on the GLP compliance statement, the final report. The test facility quality assurance unit must audit the data for the certificate of analysis from the supplier to be compliant. And last, test facility management must approve the use of an analytical reference standard that has not been characterized under GLP for test substance characterization study to be compliant. Pick up and choose.
So far we have mostly 2 and then 1 number 4 chosen.
Okay, so 2s and 1, 4.
Mm-hmm.
Let's give it one more second. And 2 is correct. It should be included as an exception on the GLP compliance statement because it's not compliant. But you know, you may think about the test facility management approval that would be sought in the very beginning, and perhaps there will be delay in the study start and a request some verification with the supplier. You cannot ask for the approval when the study is completed. That's like, okay, we deviated, but if someone approves the deviation after it happened, then we should be okay. Then that does not work this way. You may have a planned deviation sometimes that happens, but all the decisions should be made before the deviation actually occurs. And there should be some documentation of the decision. So anybody would like to talk more about why 4 was chosen as your answer?
Triana, do you feel comfortable speaking of that?
Oh, me? Sorry. I originally selected that due to not reading the question correctly, to be honest.
I know. We all do
So
that.
I think I just fell for the trap.
Yeah, it's funny because as people were putting number 2, I'm like, my God, they're reading faster than I am. Or I hope they're reading right, you know, all the questions, because that's my thing. I answer it before I read all of them, and then or you know, or don't read it you know, carefully.
Yeah.
But yeah, so that's all of us. So that's not gonna help us learn any other thing.
No, but it's just like it's a nice reminder, right? To always read
Yep,
the question and the answers correctly.
exactly. Yep.
Yeah, I mean, always read all the answers in the first place, not just in yes, this sounds right, and quickly mark that one, because there might be some later answer that would be maybe even more appropriate. So first of all, read very carefully the question. Take your time for that. Read very carefully every single answer, and only then take a decision. Do not, you know, sometimes we do this quick read and we may overlook a word like not, or some other important thing that changes the meaning to the opposite. Let's take the next one. Equipment. A sponsor company conducts a field efficacy study. The study personnel arrive for the experimental start, and the quality assurance unit arrives for the in-life audit. All required equipment is shipped to the site to coincide with the testing. So it's right there coming. It's not that it was there and it had been delivered already and there was enough time to check everything and so on. It's just happening all at once. And the GLP balance required for weighing the substance arrives at a field site damage beyond repair. Not good. At the same time, a second working non-GLP qualified balance arrives safely, intended to be used for development. What should the quality assurance recommend? Again, one option is to be selected. The study director proceeds with the study performing a daily calibration of the working balance using GLP certified ways with proper bracketing. 2. The study director proceeds with the study performing a daily calibration of the working balance using GLP certified ways with proper bracketing, documents the use of the unqualified balance as a GLP and a protocol deviation in raw data, and brings the deviation forward of the compliance page of the final report. Sometimes we think that the longest report answer must be the correct one. It's not always the case. 3. The study director amends the protocol to include the use of a non-GLP balance. And finally, 4. The study director proceeds with the study but documents the lack of using a GLP qualified balance as a deviation in the raw data and on the compliance page of the final report, and includes an impact assessment.
This is a hard one. I have to say this is a hard one. We have mostly 2, and we have one number 4. Oh, I'm sorry, 2, 4. You have 2 people who voted for 4. Oh, 3, 4. 4, 4. Now it's getting now it's changing to mostly 4s. Let's see. I have to count now. Let's see. 1, 2, 3.
Any more numbers coming?
No, it's mostly 2, and we have about maybe 5 people who voted for 4. Let's see. 1, 2, 3, 4. 4 people voted for 4. Everybody else
Okay,
voted for
so-
2.
So let's see the correct answer. Those who answered 2 chose the correct one.
It's a tricky one.
It is, and actually, if you see 4, it's practically all included inside, and also a good part of practically all one answer is included in number 2, and you have something in addition. And I confused you by saying not always the longest answer is the correct one, which is true. Sometimes it is, sometimes it's not. So just take it. This is the longest one, so that must be the right one. Like it does not apply automatically. Sometimes it does, but don't take it as a rule. So why? This is the most correct answer. So yes, we can proceed. It's the important thing. There should be the daily calibration because it's not a qualified equipment. So we need this calibration. We need to document it. And then there is definitely GLP and protocol deviation. And it should be in the final report, and it should be made prominent and not, hiding somewhere in the corner in the last line and in a small print. That would not work.
Yeah, this is definitely very tricky because I know that it kind of confused me with impact assessment. That's definitely the one. But my thought that the reason number 2 is correct is because it gives a GLP deviation and also says what did they really do, even though they really did a GLP deviation. So what that means is that you can't just say GLP deviation and then walk off and then be done with it. You need to say, okay, we did a GLP deviation, but there's going to be a lot less impactful because we still use a GLP certified weight sets. And we use a bracketed weight and all that stuff and document it properly. And so that's the bigger aspect in here, but this was a trick one. Yeah,
2, if you consider everything that would be finally at the final report, it gives more confidence in the data that despite this deviation, there is very unlikely to be some devastating impact. Because
but number 4 also talks about compliance page though too. So that number 4 also talks about, it says it's discussed in the report.
Yeah, but it does not say anything about the daily calibration, which is for me the important missing part. So have you done that? Have you done it just in the beginning, or have you really done it before and each and every use?
Yeah, so that's the only difference, that when you have a GLP deviation, it matters to explain, okay, you have a GLP deviation, but how did you try to mitigate that GLP deviation? So they can determine, was that a huge one that was going to impact the study, or then, oh, well, yeah, it's fine. They still did all the best they could. The data is still reliable and it has integrity, so we can still accept the GLP deviation.
Exactly. Expired feed. Shipment of animal feed to be mixed with the test materials was received. Upon completion of the study, it was noted that the feed had expired during the study. Which of the following is most appropriate? You can select one. And someone would be so very kind and mute your microphone, please. Reanalyze the feed and retain a sample. Invalidate all studies using the feed. Acknowledge the deviation in the final report. Dispose of the remaining supply and document the amount. Some answers are rather funny. So
yes. So we have we have mostly 3 and then and 2.
Okay.
Yep, continued 3 so far.
So let's take a look. Acknowledge the deviation in the final report is correct. And I would say, if your risk assessment says that there was a really disastrous impact, then you might be choosing 2. But I would not do it without the impact assessment, because you might be, pouring out the baby with the water after the bath. So that might be a kind of overkill, which is not needed, like invalidating all studies using the field, maybe quite a good number. So, someone would like to continue the discussion.
Yeah, Camilla, I had only written 2 because I thought the feed was the test material is mixed with the animal feed, right? So doesn't mixing an expired feed with the test material, kind of impacts the test item integrity. So that's what I felt. That's why the entire study should be invalidated, I suppose. Definitely deviations will be filed, but the impact would be, really severe, because it has actually impacted the integrity of the test item or the test material. So that's the reason I've put 2.
I understand, but that something passes the expiry date does not mean that it immediately changes on that very moment, changes the chemical and other properties and composition and all that. Even if you take it, I take medication. Okay, I have medication. It says the expiry date is 5/2024, meaning the last day of this May, it's going to expire. In reality, it does not mean it cannot be used on the 1st of June. And typically, you have the shelf life, which is shorter than the stability data. That's how it works. I had discussions with my pharma friends because I was interested to know that. So yes, they may have a stability data for perhaps 8 months and everything is perfect. And the shelf life is 6 months. So still for continuation of the time, if there is no big change in storage conditions, it's still good. I'm not advising anyone to do that, but if there is a shortage and I have my expired medication at home and I can't get a new package, and I know this piece of information, I will just keep using it until I use it all. And I did it many times like that and nothing happened. Of course, you cannot do it for like expired 3 years ago and now I am going to take it. No, that's not going to work. But you may actually do a couple of things before you decide, and this is just a theoretical discussion. Before I would invalidate the results or the studies, I would reanalyze the feed now, seeing, okay, is it the same, or something has changed? So what is the impact if something has changed? That might be a good reason to invalidate. If nothing has changed in the composition, there might be no effect. So
yeah, maybe filing the deviation first is the first step. Filing the deviation is the first step. Then probably going ahead with the reanalysis and then supporting your validity of the set. That should be
exactly
good. Got that.
Exactly. Yeah. Very, very good. And if you think that something like that cannot happen in clinical, let me tell you a short story. It can. And I remember a study that was conducted between 2009 and 12, 13, something like that, in breast cancer patients, phase 3. And there were changes in the regulation of one of the countries, and the sites were already about to be initiated. They had the drug, the supply, everything was there. And the regulatory authorities said, no, we just changed the regulations and you need to resubmit. And it took another year before the final approval. And somehow nobody checked the expiry date of the study drug. And the company, although it was a big pharma, did not have, apparently, any kind of warning system or tracker that would start, flashing the red light saying, but you have an expired medication at that site in that country. The pharmacist did not check that. The monitor did not check that. The investigators did not check that. Of course, the patients, I don't expect them to check that. And right from the beginning, for 10 months, all the patients at that site were receiving expired medication. And when it finally was discovered by the manager of the CRA, she was doing a co-visit with the CRA, then the whole hell exploded. We did that. We asked a lab to do the analysis. Just that was the only thing that we could imagine to see how bad it really is. Imagine the headlines like, okay, our patients were dying in this and that study of this and that company because they were given expired medication. But the medication was still perfectly okay. There was absolutely no change in the composition. Everything was the same as it was just received. But of course, it was very disastrous because this is reportable to the authorities and the patients and their family members and the patients, which happens in advanced breast cancer, passed away already. So that's something you don't want to live. But it may happen, and it tells us we need to check not only before the study, but we need to keep checking during this. It's not just, there might be a delay, or there might be a change. Sometimes it happens that we move it from one place to another, and it's not the first in, first out rule that tells us what to take first and what to leave for next time. And finally, we find out that something in the meantime expired. So having a good QC process in this case, and making sure that everything what is used and every single day of the study is fit for use, it's very important. And take an action. If I see it's expiring in 2 days, but the study is going to last another week or 2 or 3, then I should not be sitting and just thinking what to do. We should know. Vendor assessment. When assessing a contract research organization for use as a test site on OECD GLP studies, which of the following is the most critical item for the CRO to fix before a phase of the study is conducted at this particular CRO? The GLP archive is not on site. There is not clear delegation from the test facility management to study director. The SOPs are not approved by test facility management. There is adequate separation available. There is not adequate separation available for the test systems. That would be the logical thing that is missing there. So what would be the most critical thing? What are the answers coming?
There are 2 and 3.
Nobody voted for 1 and nobody voted for 4.
Well, we just got 1 for 4.
Okay.
Yeah, mostly 2 and 3. And
okay, so let's take a look. And Anita, what's your vote?
My vote would be 3.
Yeah, mine as well. Because
I hope I'm right.
yes, you are.
Oh, good. That would have been embarrassing.
I know. So why? We are evaluating, we are doing selection and qualification process. At this moment, the delegation to the study director is not in place. Does not mean that it's not going to be done next week. That's not a huge, lengthy process. It's quite a simple and short one. So they may be perfectly ready. Perhaps they have just appointed a new study director and the paperwork is in progress. But the fact that they don't have the SOPs approved by the management, that can mean a significant delay because perhaps it needs a review and that it needs the formal approval and the SOPs is not just one. So that will take time. Archiving. The CRO utilizes a chromatography data software system to analyze samples and generate electronic data for test substance characterization and storage stability studies. Prior to study completion, the study director prepares a duplicate copy of all e-data onto a DVD or CD. We had this last time, I think, which is audited by the QAU. The study director then deletes all electronic data from the system and archives the CD, DVD with the paper raw data at study completion. Which of the following statements is correct? Metadata do not need to be retained with raw data. The transfer method to CD must be validated and documented according to facility SOPs and must be in a human readable format. The original e-data may be copied to the media, but cannot be deleted from the system. And gold archival DVD media must be used to copy electronic data.
So we have I believe we have all twos. I think this was a repeat out as well, if I remember correctly.
Yeah, I am quite sure we had it last time, and indeed, it is correct. The transfer method must be validated and documented. There should be SOP in the first place, how to do that. The method must be validated. There should be the documentation. And after the transfer, I should be able to read it, open it on the CD or DVD and read it again. If I can't, then it's of no use at all, right? I can just throw it away. And of course, it's not the media that I would use to copy my favorite song and put it in my car player. I still have one. But it should be archival standard, which is different and has certain requirements depending on the expected number of years to be stored. So there are different classes. And still with archiving. Review
Someone says, I guess they also believe that this is covered in part 11 as well. So
yes, indeed. That's one of the things that it would lead us, any kind of metadata, any kind of audit trails that should go together with the data and should be again readable, and should be readable not only today or tomorrow, but during the whole archiving period, meaning if it's long, then it needs additional considerations. But we have just a couple of minutes left, so let's not talk about the challenges of digital preservation. That's not a part of this topic now. Review of archival receipts for original data, and the first report for a GLP study shows receipt in the archive 5 days after the report was signed. What should be expressed to the study director and management? This is acceptable practice according to GLP as long as the SOP on archiving says 5 days is acceptable. Issue a QAU finding and a phase inspection citing the gap in time of archival for the origin data as it is required to be archived prior to signing. Test facility management issues a memo approving the delayed archiving of the raw data and the final report. There is a gap in GLP compliance. The final report and data should be archived on the day the final report is signed, or data could be archived when completed before the final report signing. Now, if you see, you have 2 and 4 have quite some similarity. So what is your favorite answer?
We only have 4.
Only have 4.
Yeah.
That's amazing. And indeed, 4 is correct. Congratulations. And we are getting to the end. So let's think we have just 2 or more questions. So study inspections should be scheduled before the test initiation date, before the test subjects are euthanized, at intervals adequate to ensure integrity of the study, at intervals adequate to ensure the study director is meeting responsibilities. It should be easy.
It's all 3 so far.
And so far, everybody is correct. In-process inspection. For an in-process lab inspection, the PA auditor notices that the tubes currently being used for blood collection contain the incorrect additive. What is the first responsibility of the audit? There might be others, but what would be the first thing to do? To write an observation report citing the protocol, to communicate the concern to the technician and notify the study director immediately, to check with the study director to see if the additive being used is an acceptable substitute, to get the tubes with the correct additive, and notify management immediately.
Everyone says 2.
Do you agree?
I
Annika?
do. I do. I picked 2 as well.
So do I. And this is a little bit difficult because you need to choose 2 correct answers, not just one, which is the easy part. So changes during the study. And you see also the scenarios. A field residue study is being conducted on peaches. The principal investigator is located at the test site, company A. The study director, chemist, is located at a project management company, company B. The sponsor is located at company C. The protocol requires 5 test material applications. The last application should be made 7 days prior to the harvesting the crop. Due to abnormally warm weather, climate change, we are talking about it all the time. However, the crop matures unusually fast. If the schedule is maintained, the fruit will be 4 to 6 days past market maturity when harvested. So the principal investigator at company A decides to reduce the number of applications to 4 so that the harvest requirements can be met. Considering the protocol requirements, which of the following are most appropriate for the protocol amendment? Principal investigator signature, company A, study director's signature at company B, sponsor's approval at company C, and QAU approval at company B. And
approval.
2 answers.
Everybody says C.
C, 2 and 3, meaning study director and sponsor's approval. Anyone disagrees?
Not yet.
It is correct, yes. The principal investigator is not the decision maker, and actually the typical process for amendment, yes, you need a study director's but you need also the sponsor approval. You cannot bypass the sponsor and just tell them, we just decided. That would not work. And
We
we
made
are-
it.
we made it. We made it 1 minute past. I hope you are going to forgive me the prolongation. Any questions? Anything you would like still? I'm okay to stay a couple of minutes more. So anything you would like to add or comment or have a situation from your daily work that you think would be interesting for others, or bring some question marks in your own mind?
Yeah,
And it
there's
seems
no questions so far.
everybody just watches the clock saying, I need to leave, I need to leave. So, okay.
Yeah.
And see you next week.
Okay. Thank you, everyone. Thank you, Camilla.
Thank you.
See you next
Bye-bye.
week. Bye.
Thank you, Camilla. Thank you.